My name is Susanna Kiwala, and I'm a software developer in the Griffith Lab at the Washington University School of Medicine in St. Louis. Today, I want to talk about our immunotherapy tool suite, pVACtools, and how it can be used for neoantigen identification and prioritization. I wanted to start off with a bit of background. I'm sure many of you are already familiar with this, but in cancer immunotherapy, we aim to leverage the immune system's ability to identify and destroy cells infected with non-self or foreign elements. Instead of the foreign element here being a pathogen that has invaded the cell, it's the tumor cell itself. In normal cells, wild-type peptides are presented on the cell surface by the MHC complex, but are recognized as self, while in tumor cells, altered non-self versions of these peptides may be presented. The goal is to identify a set of these mutated peptides and delivering them to the patient to stimulate an immune response against the tumor. Predicting which new antigens might be presented by a tumor cell and selecting the ones to include in the vaccine is a complex process with many criteria to consider. I noted a few of these here on this slide, but this is by no means meant to be an exhaustive list. To tackle this problem, we have developed pVACtools, an ensemble suite of complementary Python and R tools for neoantigen identification and characterization. First up is pVACseq, which is used for predicting new antigens from missense, in-frame indel, and frameshift variants. As inputs, it takes a VCF file of somatic variants, annotated with consequences by web, and optionally a phased VCF of somatic and germline variants for identification of proximal variants that will affect the mutated protein sequence. pVACview is used for predicting new antigens from gene fusions. As inputs, it takes annotated fusion calls from either Ariba or AG Fusion. It also optionally takes a Star Fusion TSV to supply read support and expression data. pVACsplice is an upcoming pipeline for predicting new antigens from splice sites. It uses cis-splicing mutations predicted by ISMB in combination with variant calls from a somatic VCF for variant-level information. pVACbind uses a FASTA file of user-generated peptide sequences as an input. Each of these 4 tools I just mentioned generates 2 output files, a hard-filtered neoantigen list, and also a report of the best candidate for each variant, each tiered by their suitability for vaccine manufacturing. These are created using parameters set by the user, such as peptide MHC binding affinity, allele expression, and TSL cutoffs, and many more depending on the exact pipeline. pVACview is an R-Shiny web application. It can be used to visualize the results from pVACseq to further review each new antigen candidate and select the ones desired for vaccine manufacturing. It also supports output files from NeoFox or user-generated TSV files, which can, for example, be used to visualize the results from pVACview and pVACsplice. The main table in pVACview shows the best new antigen candidate for each variant. This screenshot just shows one example variant to demonstrate how pVACview displays metadata for each variant, as well as prediction data for the best peptide and the best transcript coding for it. pVACview also shows the tier assigned to each candidate and allows users to select a final evaluation for each candidate. Colored heat map and bar plot cell backgrounds and colored cell borders make it easy to interpret the data. For candidates not sorted into the past tier, red borders visually highlight the attributes failed by the candidate. Users are able to further investigate each candidate by clicking the Investigate button on the right of each row. There they can see, for example, information about reference matches. This shows details about where in the reference proteome a match was found. And they're also able to see whether a candidate's mutated amino acids are located at an anchor position, which may impact T-cell response. They can also see violin plots that show a per algorithm visualization of the predicted binding affinities of each peptide to check for concordance between algorithms. And these are just some of like a few examples of the detailed visualizations available in pVACview. For a demo of this tool, please visit pVACview.org. The last 2 tools I wanted to highlight are pVACvector and pVACpeptide. Both can be run after selecting a target list of neoantigens. pVACvector is used to generate a single DNA vector sequence by combining the shorter neoantigen sequences in an order that prevents novel junctional peptides. Lastly, pVACpeptide, which is currently being prototyped, aids in generating pools of synthetic long peptide neoantigen sequences. pVACtools is designed to be used on top of a well-established variant calling pipeline and only requires a few additional pre-processing steps. This approach allows for flexibility in terms of variant callers, filters, validation methods used. For users desiring an end-to-end solution, we have also developed a VIDL pipeline that starts with raw sequence data and produces neoantigen results from pVACseq and pVACviews. pVACtools is easy to run on the command line. It only requires a few arguments, but it also offers a large set of optional arguments to customize your run. Some of its features are support for multiple class 1 and class 2 binding affinity, elution, and immunogenicity algorithms, support for non-human data, prediction of peptide stability and cleavage sites, and many more. pVACtools is open source, open license, and easy to install. Version releases are available on PyPy, GitHub, and Docker Hub. Documentation is available at pVACtools.org, and an intro course can be found at course.pVACtools.org. With that, I would like to acknowledge and thank the Dr. Griffith, the members of my lab, especially the members of the pVACtools team, and all of the funding agencies that have made this work possible. Thank you.